And next slide I'll show you a color-coded structure of the transcribing complex, which will immediately follow. So it was known when we did this work from biochemical studies done over the years that some 15 base pairs of the DNA double helix are unwound in the form of a transcription bubble at the active center of the polymerase molecule engaged in transcription. It was furthermore known that some 8 to 9 base pairs of RNA, DNA hybrid are centered within the bubble, with the growing end of the RNA adjacent to the catalytic magnesium ion of 3 residues from the single-strand, double-strand DNA junction. In this picture, and all of the slides to follow, the strand of the DNA that directs RNA synthesis is blue. The RNA is red, and the other strand of DNA, the non-coupled strand, green. Now, the next slide is a movie. It is made of only 2 frames. The first frame is, each frame is an actual crystal structure. The structure I already showed you. This is the structure of the enzyme alone. Viewed down the central channel, this differs only from the previous picture in that the protein is almost all in a single color. You'll understand the reason why part of it is different colors. The second frame of the movie is the structure of the polymerase in the form of an actively transcribing complex. So what you understand from this, the DNA in blue and green and the RNA in red do indeed occupy the central channel of the transcribing complex. And now you will have noted that this massive protein element that receives contributions from 4 different subunits, which is colored with gold, swings some 30 angstroms over the DNA and RNA in the course of forming the transcribing complex. And for that reason, we refer to this as a clamp. It's colored gold, and it is important to mention here it will serve as a landmark to the larger, more complex structures that I will show you in the slides to follow. Now what I will do is rotate around and then remove some of the protein that is in white. And this gives, I think, the best view of the course of the DNA and RNA through the transcribing complex. So here, both protein and DNA have been modeled into the electron density derived from X-ray diffraction analysis. And the result shows the DNA double helix enters in this direction to the right, unwinds 3 residues before the active center, after which there is a sharp bend in the template strand, in consequence of which the next base is flipped 90 degrees and points down towards the active center to read out the transcription. In this structure, that DNA base is paired with the ribonucleotide just added to the growing RNA chain. There are 8 more RNA DNA hybrid base pairs seen in this particular structure. Now you will have noticed that the axis of the entering DNA of the helix is at nearly right angles to the axis of the exiting RNA DNA hybrid helix. And that is because of what we call a wall of protein density that blocks straight passage of the nucleic acids through the polymerase cleft. That wall of protein density will be colored blue in some of the slides to follow and serves as another landmark for the structure. What I will do in the remaining time, first in the next few minutes, tell you about 2 examples of interesting insights derived from the study of such structures pertaining to the fundamental mechanism of transcription. And then in the remaining time after that, I'll speak about the interaction of the polymerase with many additional proteins responsible for the initiation and the regulation of transcription. Now the next slide is the same as the one you see here, except it is in a surface rather than a ribbon diagram. So in the surface representation, same view, the DNA enters from the right, the RNA DNA exits at right angles. But in this surface representation, where the cut surfaces where the protein was removed to expose the nucleic acid are shown in gray, you can appreciate there is a hole through the nucleic acid from the bottom up to the active center. Ribonucleoside triphosphate precursors of the RNA enter through this channel. All four combine at a so-called E or entry site. This we know especially from the previous work on biochemical and genetic of low-carbon polymers. Only the ribonucleoside triphosphate correctly matched for pairing the coding DNA, the coding base and the template DNA strand can rotate into the addition or A site where the growth of the RNA chain takes place. One of the two insights regarding the fundamental mechanism that I would like to mention that derives from work along these lines concerns the basis for the fidelity of transcription. That is, after all, the essence of transcription, the accurate readout of the genetic code. The fidelity of transcription is very great. The error rate is about 1 in a million. That cannot be explained on the basis of base pairing alone, which would contribute only 2 to 3 orders of magnitude at most. The basis for the fidelity of transcription was not immediately apparent. It could not be seen in the structures that I've just shown you. Work done in the subsequent years that achieved an extension to higher resolution of the structure that revealed the basis for the fidelity of transcription. It resides in a feature that we call the trigger loop, seen here from multiple structures. In most cases, in telomerase, in the absence of nucleoside triphosphate precursors, the trigger loop was at a distance about 30 angstroms from the active center, and in some cases, disordered. Only in a structure that included an appropriately matched nucleoside triphosphate paired to the coding base would the trigger loop be seen in the orientation that is shown here in purple. And in this orientation, the trigger loop is in position to interact directly with the entering nucleoside triphosphate, making interactions with the base, the phosphates, and also indirectly with the sugar moiety, the entering nucleoside triphosphate. It is by virtue of these interactions that the trigger loop is the selection of the correct nucleoside triphosphate, the growth of the ribonucleotide polyribonucleotide chain. I can explain later exactly how these interactions lead to the enable the high fidelity of transcription, if anyone would like to ask. But in the interest of time, let me go on and mention one other fascinating insight concerning the fundamental mechanism before I can to the larger problem of the initiation and regulation of transcription. So another puzzle about transcription beyond the issue of fidelity is how do you separate the RNA made alongside the DNA template in the form of the DNA-RNA hybrid for its release into solution. An RNA-DNA hybrid is very stable, and it was not at all apparent at the outset how the RNA could be peeled off the complete DNA strand. That was only apparent some years later from the structure in which the process could be observed to occur. In that structure, what we could see, and now I show you only the portion of the DNA that lives at the upstream end of the RNA. So let me just back up the stage. In these slides, I have omitted almost all of the protein. I focus only on the nucleic acids in the vicinity of the active center. In the one that I will describe in a moment, I narrow the focus even further and I show only the portion of the RNA DNA hybrid that So in this region, what we see, in fact, starting at the active center and counting up to 7 residues away, there is a standard RNA-DNA helix. The bases are coplanar. They are separated by a distance appropriate for hydrogen bonding at 3.4 angstrom. But further distant, 8, 9, 10 residues from the active center, the bases are progressively splayed apart and separated from one another by a distance of 10 or 12 angstroms. This is due, we can see in the structure, to the intervention of 3 protein loops. One that we call rudder, which interacts with the template DNA, maintains its separation from the RNA, and another called lid interacting with the RNA. A side chain of a residue in the lid actually serves as a kind of rudder, as a kind of, sort of the word, a kind of divider to drive apart and force the separation of the strand. The remaining loop, which we call a fork loop, does the opposite. Positively charged lysine side chains of the fork loop contact phosphates on the sugar phosphate chains of the template RNA strand to stabilize the base pairs at positions 7 and 6. And this way, to prevent the unraveling that is enforced by rubber lid from extending all the way to the active center, disrupting the DNA RNA helix and inhibiting the process of transcription. Now let me turn in the remaining time to the role of the general transcription factors in promoter recognition, in the initiation of transcription, and then finally to the regulation of the process, which I have not touched upon at all at this point. I'll show you only one example of studies and of general factor interaction with RNA polymerase. That is the first on the list, factor B. And then I'll show you a recent, still unpublished result that encompasses the entire constellation of proteins. What you see in this slide is the result of a co-crystal structure of the RNA polymerase with general factor B. Such structures were solved by Bushnell in 2004, by Liu in 2009, and also by a former colleague, Patrick Kramer, in his own laboratory in Munich, also in 2009. This is the polymerase that I showed you before. Viewed from the top, here's the cleft through which DNA enters, passing to the active center. This again is a surface representation rather than the ribbon diagram I presented before. Here is the clamp in gold. Here is the wall in blue that blocks straight passages of nucleic acids through the cleft. The structure from this co-crystal determines the factor B, is shown here in red. Now, years ago, Burley, Sigler, and their colleagues determined the structure of a small protein DNA complex made of a fragment of this factor B, a fragment of the so-called Talobox binding protein, component of factor B. And